welcome back to my channel. Today I will explain about the detection methods of anthrax. In my past video I explained about general information about anthrax symptoms, what's anthrax, who was the first person who found that anthrax detection methods. But now I will explain the detection methods by using pictures. First, anthrax detection. We have six common ways to detect anthrax. First, blood Giemsa stain, Giemsa or bright stain. Blood or oedemal fluid culture. As I said, we aspirate sample is aspirated by needle. Using needle, we aspirate fluids from the lesion that are called in cutaneous anthrax. That the fluid is called oedemal fluid. And we say macrophages are presented in the fluid. Why? Because when the lesion happens or occurs, the macrophages go to the surface of skin to fight the bacteria, to prevent it getting into the bloodstream. We as I said, blood or oedemal fluid culture. We have biopsy culture. Sometimes lung biopsy is taken, and sometimes spleen. It depends on the case. Ascoil test is an antibody test. Post-mortem examination. As I said, in some countries, post-mortem is illegal. And we have clinical symptoms. We spoke about the symptoms. I will not explain the symptoms in this video because as I said, respiratory distress, loss of appetite, lesions and paracute lesions on the neck and the abdomen, and bleeding from the body openings, and death at the end. As I said, let's give a review. What are the six methods? Blood Giemsa stain or light stain, blood or oedemal fluid culture, biopsy, Ascoil test, post-mortem examination, and clinical symptoms. Here, the first thing, we have blood Giemsa stain. We have some different stain, not just blood stain, we have some different variation of detections. Here, guys, we can see here rod, but as I said, the shape is rod, bacilli, rod shape. It's gram-positive rod-shaped bacteria. And it's capsuled. As you see here are the bacterias, long, rod-shaped. In blood cells, we have the bacteria. These are Giemsa stain. And and this is the result of blood smear Giemsa stain. The bacilli is detected. Here is the bacilli, short bacilli. Now in this case we have short bacilli, but anthrax is a long bacilli, rod-shaped, capsulated, spore forming. How the culture is done? Bacillus anthrax culture is blood agar and nutrient agar are used at the pH of 7.5 to 7.8, and incubated, and the temperature should be 37 Celsius as other bacteria. Colonies after growth, it will take about 5 to 12 days, in rare cases 2 days to grow. But and the colonies, when they grow, the colonies are non-hemolytic with gray color. The colonies are gray in color. Note, we have a note here. The note is sometimes HCO3, we mean bicarbonate. This is not correct, but I was forced to use a short form because the slide is just one slide. Sometimes HCO3 bicarbonate agar is used, but the best choice is blood agar, as I said, or nutrient agar. But blood agar is the best. Here is the picture of the colony. As I said, gray, gray colony, large gray colony. But I have a note. When you do a gram stain for for anthrax, mostly for bacillus, acid fast stain is done. But when you take a colony, when you pick a colony using a loop, when you pick a colony of anthrax, the colony is sticky, more sticky than usual. One of the ways to detect the colony. The colony is called medusa head, gray medusa head colonies on blood agar. This is the picture. Gray colonies are detected on blood agar. PM or postmortem. In postmortem, the spleen is enlarged and lesions on the lung. It depends on the type of anthrax, but all the anthrax mostly the spleen is enlarged. That's why sometimes it's called splenic fever. Sorry guys, I'm a little bit sick. It's called splenic fever due to the enlargement of the spleen, and lesions are detected on the lung in inhalation anthrax, and sometimes intestine lesion and gastrointestinal anthrax. Lesions are detected on the intestine. But let me say you something else. You should remember this. Let's review this. Postmortem in most countries are illegal. Anthrax lesion. Here you can see, guys. Necrotic lesion, you see, depends on the stage. This is a late stage of anthrax, D. In A, we see flatulence, a dead anthrax infected animal that's infected by anthrax. Flatulence is detected. Flatulence in A, we see a dead animal and flatulence of anthrax. Here we see bleeding in the nose, as I explained, bleeding in the body openings of a cow, such as nose, vagina, and sometimes eyes. Here is the bacilli rod shape gram stain. Those are in D, this is paracute anthrax. Here we have acute anthrax, those necrotic lesions, and this is the lesion on the neck, and in the back of the leg. We have another test that's used, it's called Ascoil test, Ascoil precipitation test. How is this test done? This test is done by taking samples and boiling it with saline, and first antiserum is added, and the boiling, for example, you have bone meal. Bone meal is added and boiled for 10 minutes with saline solution. After that, it's added to the serum antibody, and the results are detected after. You see, here in the left we have a negative result. In the right, precipitation is detected. It's a positive result. As I said, sample is taken, such as meat or mostly bone meal is taken, and the bone is break down by a specific machine, and after that the bone is boiled with saline solution, and the test is done within 10 minutes, and the antibody is detected, and the results are detected here. As I said, in the left we have a negative result, and at the right we have a positive result, because precipitation is detected. You see, precipitation is detected. We have biochemical test results of anthrax. Biochemical test results. For every bacteria, we know we should do biochemical tests. For example, we have catalyze test, gelatin hydrolysis test, gram staining. The first test is done is gram staining. So, let's go. As I said, the bacteria is capsulated. Catalyze test is positive. gas is negative. No, we don't have any gas production. As I said in the past video, TSI test is done, no gas production. But in bacteria such as E. coli, we have gas production. Gelatin hydrolysis positive, so it means it's able to hydrolyze gelatin or break down gelatin. Gram staining is gram positive. What's the difference of gram between the gram positive and gram negative bacteria? The gram positive bacteria have more peptidoglycan peptidoglycan, but the bacteria that are gram negative have less peptidoglycan. So that means gram-positive bacteria have thicker cell walls, but gram-negative bacteria have thinner cell walls. Hemolysis, as I say, they are the colonies are non-hemolytic. Indole test is positive, and the bacteria is non-motile. Motility test is negative, so the bacteria is non-motile. But for example, E. coli is motile, motility positive. Sorry guys, I'm a little bit sick as I say. Nitrate reduction is positive, so it means the bacteria is able to the bacteria is able to produce nitrate. Oxidase negative. Negative, the oxidase is negative, and pigment is negative. We don't have any pigments in the bacteria. Oxidase test, how is it done? We bring a clean sterilized petri dish, glass petri, yeah, and we add a filter paper. We put a filter paper in the glass, and we hold a colony using a stick, because if you use a metallic loop, it will give you false results. So oxidase test is done by using a stick. We take a single colony and we add the reagent on the paper first. We add the reagent on the filter paper, and after that, by using a stick, and of course we should work near the benzene burner or alcohol burner to prevent contamination. But not for oxidase test. For aseptic technique is benzene burner is used. So you take, for oxidase test, you take a colony using a stick. We should not use a metallic loop. Using a metallic loop will give you false results. And you add the reagent, and it's called oxidase reagent. You add it on the filter paper, some drops of the reagent on the filter paper, and you will, using the stick, you take a colony and you put it on the drop and mix it using the stick. Mix it. The results, if blue color is appeared, so it means its blue-like color is appeared, it means the test is oxidase positive, and the shape is rod shaped. So thank you guys for watching. Hope you get benefit.